So we noticed that for day one, for one of the treatment groups that was exposed relative to control, we saw that there were some genes here. About 155 genes were significantly differentially expressed. 108 was upregulated. But of course, at day 5, we're seeing that the number of genes that were downregulated were really high. 1,502 were significantly downregulated. What we now wanted to see is what's going on here in these genes that could maybe help us understand the phenotype. So what you see here for day 1, where we see them gain more weight, we do see upregulation of genes. For instance, here is D, which is structural and cytoskeletal related genes. So what that means is they are changing, shedding their cuticle a little faster as they're growing bigger. So probably that's what we're seeing here. All of the genes, some 20 of them were all related to that. Of course, we do still see some increased metabolic gene relative to control that we see here. But now when you come to day 5, you would notice there is a global downregulation of most of the genes, especially when it comes to energy and metabolic related genes. And it makes sense because if the larvae is preparing to pupate, usually they stop eating, they slow down the metabolism to conserve energy in the pupation state. So we feel that this evidence kind of supports the fact that they are getting to the pupation state a little faster than control. So with that said, we also tracked how much food they were eating. We saw that the low treatment groups, day 1, day 2, higher food consumption. We support what we saw in our soybean leaf exposure experiment. We also noticed that day 3, day 4, nothing much was happening. This insert here shows the prepupation at day 5. Again, we're seeing that by day 5, they were already prepupating, but the controls didn't have any prepupation at that day. So again, this supports our hypothesis that the chemical is making them move through their life cycle a little faster. Right here, we now looked at the average time it was taking them to get to pupation. On the average, the ED50 here is telling us how much, which is the time it takes 50% of the population to pupate. We're seeing it's taking 7.5 days for the control, but all of our treatments are way less than 7.5. In fact, this very low treatment group was somewhere around day 7. So they are pupating almost half a day faster than the controls. You come here also, we also see that there is slightly faster time to get into adults that we're seeing here. So what this is telling us is the chemical, because it's making them gain weight faster, they're pupating a little faster, and of course, they are transitioning through their life cycle a little faster. And I try to simulate that. I try to do the computation. If they are moving through their life cycle just 0.5 days faster, in 3 years, they would have the population explosion is going to be 6 times in the treatment group relative to control. So that said, this could be a potential question that we'll be looking at. All right. So the study here is showing PFBA has the potential from this study in the lab to stimulate the growth of insect larvae. So if that is the case, could that be something that IPM strategists should also be thinking about? Because these chemicals are in the environment, and most times for PFBA, nobody really cares much about them from human health perspective. So the question is it something we should be thinking about? So of course,